Our next speaker this afternoon is Dr. Claire Jardine from the University of Guelph Department of Pathobiology. And she'll be talking about Baylisascaris in raccoons in Ontario.
Thanks, Andrea, and thanks very much for this opportunity to talk to you today about some work we're doing looking at Baylisascaris procyonis, also more commonly known as the raccoon roundworm. So I'm just going to start today by just telling you a little bit more about the definitive host of the parasite, so raccoons. They're common omnivorous medium-sized mammals. Most of you are probably familiar with them. Their diet is quite interesting. It's really determined largely by the environment that they live in, and they will really eat almost anything that they can. And some of you may have seen them in your garbage, for example. They often live at very high densities and in close association with humans. And researchers have recorded densities as high as 94 raccoons per square kilometer in some urban areas in Ontario. Raccoons are known to harbor a number of zoonotic pathogens, including Leptospira and Salmonella, for example, in addition to Baylisascaris. And this high raccoon density, combined with their close association with humans, has led to concerns about their role and perhaps an increased risk of disease transmission from raccoons to humans, particularly in urban areas. So the prevalence of Baylisascaris in raccoons tends to be quite high. There's some reports from the mid-1980s where the prevalence ranging from about 43 to 57% in Ontario and Quebec. It's considered to be an emerging zoonosis. There seems to be an increasing number of reports of human disease from endemic areas, and there's some evidence of spread to new areas in recent years as well. So I'm just going to go over the life cycle just in case anyone isn't familiar with it. So raccoons are the definitive host for Baylisascaris. There, and that's where the adult worms live inside the intestinal tract of raccoons. And just to give you an example of what that can look like, so here's a heavy burden inside the small intestine of a raccoon. Although usually we don't expect to see any health consequences from a Baylisascaris infection in a raccoon, very heavy infestations can lead to intestinal blockage in some cases, but that's not common. So the female worm releases eggs, which are passed in the feces, and an infected raccoon can shed, they've estimated approximately 20,000 eggs per gram of feces, so they're pretty heavily loaded with eggs. So the eggs are then in the environment, and they're not immediately infective. It takes about 2 to 4 weeks for the eggs to embryonate and for the larvae to develop. But once the eggs are larvated or embryonated, they can remain in the environment for years, and they're very difficult to kill. So raccoons can become infected by different routes, but often they're infected directly through ingestion of these embryonated eggs from the environment. And other species of mammals and birds can be infected also from these embryonated eggs in the environment. And these are known as either intermediate or paratenic hosts in this case. And the worms don't develop in these animals. The larvae move around inside the body and encyst in various locations, most notably the nervous system. And this just shows some larvae in the brain of a gray squirrel, just as an example, so that you can see that they cause quite a bit of damage. Okay, so then the life cycle is complete in this case when the raccoon eats one of these paratenic or intermediate hosts. The larvae develop into the adult worm. And it's interesting to note, and important to note here, that dogs can also serve as definitive hosts for Baylisascaris, and that has important public health implications that should be considered. So you can see, so there's 2 routes of infection for raccoons. It's generally believed that juvenile raccoons become infected through the ingestion of these embryonated eggs directly from the environment, whereas older raccoons typically are thought to become infected by ingesting these intermediate or paratenic hosts that have the encysted larvae. So where do humans fit into all this? Well, unfortunately they can accidentally ingest these embryonated eggs, and then In humans, the larvae can migrate, much as they do in the intermediate host, and they can go to a wide variety of tissues, including the brain, but also liver, heart, lungs, and other tissues, and you get visceral larval migrans or ocular larval migrans, similar to what you see with toxocariasis. But in contrast to Toxocara larvae, Baylisascaris larvae continue to grow inside the human host, and the tissue damage and signs can be often very severe because the Baylisascaris larvae continue to grow. They move around a lot. They don't readily die. And so, unfortunately, diagnosis of this disease in humans is often made through autopsy specimens, but it can also be made through serology, and occasionally they're able to identify larvae in biopsy specimens. So it's thought for humans that young children, are at greatest risk of infection, and this is really because of their propensity to put things in their mouth, including dirt, and perhaps their poor hygiene habits. However, really any human that's in contact with raccoons or raccoon feces is potentially at risk. So we felt that developing a better understanding of the epidemiology of Baylisascaris in raccoons is important. It would help us identify factors that may be associated with increased risk of exposure to Baylisascaris in the environment. So the effect of various factors, including land use, demographic factors of the raccoon, such as age and sex, and season, on the prevalence of Baylisascaris in raccoons have been looked at, but there really are no clear conclusions from the studies that have been done to date, and the results are actually quite conflicting, and I've just put a few examples here. So a paper from 2008, Page et al. Found that the prevalence of Baylisascaris in raccoons in urban areas was lower than in rural areas, despite having high raccoon densities in these urban areas. And they hypothesized that these urban raccoons may be relying more on human related food sources, so that this transmission route was reduced. So they hypothesized that led to a decreased prevalence in these urban areas. However, other researchers have found no difference in the prevalence between urban and rural areas. And in fact, some researchers have found that the prevalence of Baylisascaris in these intermediate or paratenic hosts is actually higher in urban areas, and this sort of suggests that there is probably greater environmental contamination of the embryonated eggs in these urban environments. So our objectives for this study, so there's a lot of conflicting results out there, and our objectives were, first of all, to provide updated information about the prevalence of Baylisascaris in Ontario. We get asked this question a lot at the Wildlife Health Centre, and it's nice to have more updated data than the information we had from the 1980s. So in addition to getting this prevalence information, we also wanted to assess the impact of land use, demographic factors, and season on the prevalence and intensity or number of worms in raccoons in Ontario. So those were our objectives. So what we did, we conducted necropsies on all raccoons that were submitted to the Canadian Wildlife Health Cooperative between March and October 2012 for this particular study that I'm reporting on now, although the work is ongoing. We recorded carcass location data, age of the raccoon, sex, number of Baylisascaris present in the intestinal tract. So we conducted logistic regression models to examine associations between the presence of Baylisascaris and land use, which we classified, here I've called it urban, but it's really urban/suburban, and then rural. And we classified this based on the amount of development in a one square kilometer area around where the raccoon carcass was found. So if there was greater than 10% development, we classified it as urban/suburban. And then we also looked at season, which we divided into two different time periods, March to June, which roughly corresponds to kind of breeding period, and then the non-breeding season from July to October, and then age and sex. We use negative binomial regression models to look at associations between the number of worms and the same variables. And we built all these univariable models first, but all variables and significant interactions were included in the final multivariable, when we were building the final multivariable models. Okay, so here's a map of southern Ontario, just showing the locations and land use classification of the Baylisascaris positive and negative raccoons that we got in. There's a total of 128 from this time period that are shown here on this map. The triangles, which you can see, represent rural areas, and the circles are classified as suburban/urban areas. And the green shapes are the raccoons that are negative for Baylisascaris, and the red, whatever, either circle or triangle, are positive for Baylisascaris. Okay, so they're pretty widely distributed, really. And the median number of worms in the intestinal tract was 3, and it ranged from 1 up to 116 during this study. So, sorry, just to go back, the prevalence based on that initial sample was 38% of the 128 raccoons that were submitted. It's a bit lower than what had been reported previously, but there was a really strong seasonal difference that you can see here. The prevalence so this figure shows the percent of Baylisascaris positive raccoons. The error bars represent 95% confidence intervals, and then the number inside the box are the sample size, and this is just based on the raw data. So you can see that the prevalence ranged from about 17% in June all the way up to 88% in September, which is quite dramatic. So what's interesting here is that there's a hypothesis that the prevalence of Baylisascaris seems to undergo a yearly cycle in raccoons, and at least in temperate areas. And it's thought that many raccoons clear infections in the fall, and we don't have the data to show that, but what's interesting is it comes around in the spring, we have a much lower infection rate, which does sort of lend evidence to support that hypothesis. So we thought, given this marked increase in prevalence in the fall, it might be worthwhile considering timing any public health messaging related to the risks posed by raccoon feces to kind of coincide with that fall time period. Keeping in mind, though, that raccoon feces are never good and should be avoided at all times, and that infectious eggs can remain in the environment for long periods of time. Okay, so this table shows the prevalence of Baylisascaris for the different groups, in case you're interested in looking at that. And then it shows the results of the univariable analysis over here. And what you can see is that the presence of Baylisascaris in raccoons was significantly associated with age, land use, and season in these univariable models. So, for example, juvenile raccoons were almost 3 times more likely to have Baylisascaris than adult raccoons. Raccoons in urban areas were almost 3 times more likely to have Baylisascaris than those in rural areas. And then, as you saw from the raw data, the prevalence is much higher in the second time period than earlier in the spring and summer. All right. So this shows the number of worms. So this is the median number of worms in that first column there of numbers, and then the results of the univariable models, and the results are quite similar, that age and season are both significantly associated with the detection of more worms in raccoons. All right. So now, we constructed the multivariable models, and we included all the variables that were statistically significant or part of a statistically significant interaction term. And what's interesting in these final models is you can see these effects down here called interactions, and I'm going to talk more about those in a minute, but they're quite interesting in this case. But just before I get to that, I just wanted to show you the results of the final model for the number of worms. And you can see that the results are really quite similar. We have these interaction effects. In addition, we do see this impact still of age on the number of worms. So the fit for this model, this particular model, looking at the number of worms, wasn't that great. So we need to, we need to be cautious when we're interpreting this model. But the first model that I showed you, looking at the prevalence of Baylisascaris, did fit quite well. And I'm going to talk about those interaction effects in a bit more detail now. So this figure shows the predicted prevalence of Baylisascaris for male and female raccoons in urban and rural areas over the 2 time periods. And you can see that the impact of land use and season on the prevalence of Baylisascaris was dependent on sex. So male raccoons shown here, if I get this right, that they are the non-colored in symbols. We always see that urban male raccoons have a higher prevalence, and it's significantly higher than what we see in rural male raccoons, okay? So that's interesting. So we do see this impact of urban, the higher prevalence in urban areas, and it could be, so we see an increased contact, people have reported increased contact rates, particularly in male raccoons in urban areas, and because animals live at very high densities in those areas, and it's thought that male raccoons may be less solitary than we at first thought. So it could be that there's increased contact rates, although we don't really know that that's true, but that could be what's driving this situation. But what's really interesting, I think, from this figure is that we don't see that same effect for the female raccoons. So the female raccoons are shown here in these colored in symbols. And what's really dramatic here is you see this marked increase in the prevalence of Baylisascaris in the late summer, fall time period, only in the female raccoons. And there's no difference between the urban and rural environments. So that dramatic difference between the sexes hasn't been reported previously, and may be related to behavioral differences between males and females, resulting in greater exposure to Baylisascaris in the late summer and fall for these female raccoons, perhaps due to their close association with the juveniles throughout the summer, although that's something we need to look into. So I guess one of the take-home messages for me from this little project was just when we look at the results of the univariable analysis that didn't take into account these interaction effects, we can see that there's a lot of variation. You get a lot of variation in your results depending on when you did the sampling, what kind of animals you sampled, and where you were sampling. So I wonder, it made us wonder if some of the inconsistencies in the findings from previous studies might be explained by the interactions that weren't taken into account when you just look at the univariable, when you just use for univariable statistics. All right, so that's, now I'm just gonna give a quick summary that we think the epidemiology of Baylisascaris in raccoons may be much more complex than we previously thought. The impacts of season and land use on the prevalence of Baylisascaris appear to be dependent on sex. Further understanding on how the transmission of this parasite differs between males and females will allow us to better understand those results. And we hope that this information will be useful to inform public health officials about spatial and temporal factors that may lead to increased risk of Baylisascaris transmission to humans. And the study is ongoing. We're sort of in the middle of a 5-year study trying to get better, more comprehensive data on this to try to test some of these ideas. All right, so I'd just like to thank all the people that assisted with carcass collection, assisted with necropsies, and especially Kirsty Puskas, who was supported by a Pfizer Summer Student Research Scholarship, and she helped to lead the summer portion of the project in 2012. Thanks.
We can take a couple of questions before the break.
Yeah, that's it. So the question is, how long is the lifespan of the worm? Because you do see that sort of loss of worms over the winter period. At least that's how it appears based on the data. I don't know exactly what the lifespan of the worm is, but that is definitely the hypothesis, that as the raccoons kind of, they bulk up for the winter and then they kind of den up and they're not doing a lot of eating and foraging at that time, and it's thought that the worms kind of die out during that time period, and then they have to become reinfected in the spring. That's the hypothesis. What's interesting with our data, in more southern areas, they don't show that, but as you start moving north, you start to see that trend, but it occurs much later than what we're not much later, but like September, October is what's been reported previously in the literature, and when we're seeing it now in July, that's sort of starting this increase. So that's kind of an interesting point. So, yeah. Yeah, that's a good question, and we've thought about that a little bit. So Dr. Smith was just asking if this increase in the prevalence in female raccoons may be related to lower body condition and maybe the impact of having all these babies hanging off you on your immune status. And I think it's a good hypothesis, I think, and it would be interesting to investigate that. What's interesting though is even the female raccoons are starting to bulk up by that time period. But I think if we could, look a little bit more carefully at some of like kind of the reproductive status and that sort of thing, we may be able to get a better handle of that. And of course, it would be great to do some sort of immune assay if we could ever figure out how to do those properly in wildlife. Yeah.
Join me in